stem of my brain. I think, Alex, we met the first time when you were a PhD student with Monica organizing a meeting in Obergurgl.
Yeah, exactly.
This is where I have the first sort of memory. And I already realized that actually there is a very smart PhD student sitting there at the registration desk.
Nice.
So it was actually quite neat to see to meet each other again over and over the years, and now we have even a joint FWF project. Alex studied physics at the University of Innsbruck. And I think you stayed probably with Monica for a brief postdoc before you moved to UK, to Martin Booth's lab. And then you returned back to Monica as an associate professor, which you are right now, which is now at the Medical University of Innsbruck. Did I say something wrong up to now?
No, broadly speaking, correct.
And I can just say that it's a pleasure to work together with Alex. I have now the experience since 4 years, and I hope that we will continue like that.
Gerd, you left out Stefan. He was also supervising, very importantly.
Stefan,
yeah.
So. I consider Alex for me as a microscopy expert, and whenever I'm running short of microscopy and optics, a Zoom call usually helps out. We collaborate on 3D single molecule microscopy, and I think this is also the topic of your presentation. Let's say the bio part will later on come by Lucas, where we apply the system in order to do fun stuff on T-cell biology. But now the floor is yours, Alex.
Okay, well, thank you, Gerhard, for this kind introduction, and thank you also for inviting me to be here and to the organizers of the workshop. Okay, so I'm going to speak about, I guess you see my screen, about localizing single molecules in 3D close to a dielectric interface. It's pretty much the same application target field that Jörg already addressed today, right? Okay. Okay, so that's the outline of my talk. So as I said, I will focus on this kind of measurement environment, localizing up to, let's say, a third or so of the wavelength close to the cover slip. And then I actually split my talk in 2 parts. In the first, I'd like to introduce defocused imaging as a pretty simple but powerful method to obtain high axial precision in SMLM. And in the second part of my talk, I just ask a bit more general question. What's actually the best, and I say it here under quotation mark, classical 3D localization approach? Yeah, classical meaning that I'm only using the tools that typically researchers have in their lab, a wide field microscope, a good standard camera, and drawing only information from the images that you actually detect. So not actually measuring lifetimes, which we have seen is super powerful, but also not playing with the excitation path, like using varying angles and so much, just using information on the camera on the detection path. And Jörg already mentioned what the problem is in microscopy. The third coordinate, the Z coordinate, is always much worse due to the PSF shape than the lateral coordinates. And that was known from early on, actually, and people have devised methods, to mitigate this. This is what I call the classical methods. And we've already heard today about biplane imaging, for instance, which works such that you split your beam after the objective lens and you slightly detune one of the planes such that you get more diversity in your images. That has been actually introduced even before the SMLM era by Raimund Ober's group to actually extend the axial working range. And later, I think the group of Jörg Beversdorf has been the first to introduce this technique for SMLM, mainly also to stretch the axial working range there, right? This is the main application. Pretty much at the same time, other techniques came up called like PSF engineering approaches. The most commonly used the astigmatism mode. We've already seen talks about this today, which basically introduces a known aberration to the system that likes to break kind of the symmetry in the PSF, and then you know about where the actual molecule is located along the optical axis. And more advanced schemes are out there, like double helix PSFs introduced by Raphael Pistun, and other shapes, saddle point, tetrapod shapes, all kind of numerically optimized using Fisher information for specific target applications. Now, what we have already seen today is that many applications actually don't need to be applied in the optical far field, right? Very often you are interested in membranes or cell compartments, bacteria or viruses that can be very close to the cover slip. And in our case, and that's the collaboration with Gerhard, it's the same, right? We have, from a purely metrology point of view, the task is to map out this interface between T cells and antigen presenting cells, which is here mimicked by a functionalized bilayer, as accurately as possible. And as you see already here, the Z coordinate is of particular importance. So we were wondering what can we do to improve as much as possible to get a robust, very precise localization method. And well, close to the cover slip, to the dielectric surface, or even better to a metallic surface, as we've seen today, you can actually use other alternative sources of information. And yeah, what Jörg presented today, which I really like, is this MIT method, which measures lifetimes that change if the molecule is over a metal, but even slightly if it's close to a dielectric like glass. Yeah, other groups have used standing wave effects that occur in metal coated surfaces as well. Then also we've seen today what's possible is play with the exponential decay or exploit exponential decay of the excitation field. If you're in turf geometry, you can either directly exploit the supposedly known exponential decay that has been actually recently been shown very recently by a group that this is possible, or to play with the turf illumination angles, and then you evaluate the brightness trends that your molecule undergoes, and then you can also learn about the Z coordinate. The exponential decay, or this evanescent trick, can be also played on the detection side. Yeah, and this is called, also mentioned by Jörg today, is called supercritical angle fluorescence, or in brief SAF, which is a very well-known effect actually. It's a near field coupling of a molecule. Yeah, whenever a molecule comes, fluorescent molecule comes really close to an interface or to a material of higher refractive index, then a part of the evanescent field turns into propagating waves. And those waves then propagate beyond the angle of total internal reflection, as you see here in these back focal plane images, right? So we have here a so-called UAF or WAF zone, where we have light that travels ordinarily, so to speak, within a circle defined by total internal reflection. But whenever we are very close, then we get a lot of light. This can actually be up to 50% of the total emission that is collected by the objective lens that travels here in this SAF range. Now, the basic idea in this effect, actually, although it's well known, and it has been exploited also very early in microscopy, in around 2000, for SMLM, it has only been used pretty late. So it was 2014 and 15 where two groups have pioneered this work. The group of Jonas Ries and Sandrine Levick-Ford have actually devised a setup to use this SAF effect in SMLM. And the idea is pretty simple. It is based on measuring the powers in the UIF zone and the SIF zone and then putting them into relation. Yeah, because this is supposedly a very sensitive ratio, which is sensitive on the Z position of the molecule. And the setup they proposed looks like that. Basically, a microscope, you split your beam. In one channel, you just have your normal molecule image. In the other channel, you block the SAF part such that you only collect UAF photons. And then you estimate the numbers of photons in both channels and you calculate the ratio. And then you end up with a curve like that, which, as you see, is pretty sensitive against the Z coordinate. And that allows you to obtain pretty high precisions. And when we were discussing our project, this was pretty neat, right, for us. What we liked about these SAF-based methods is that they're super simple. Yeah? We don't even need to know what is our turf illumination angle, right? There can often be problems with turf illumination fields, even if you have some dust on the cover slip and so on. It's pretty error prone, I would say. but on the detection side, it's much more robust. Yeah. And the only thing we supposedly need to do is split the beam, block a little bit of light, and then estimate intensities, which should be pretty straightforward. The first thing that we actually planned to improve on this SALM or Donald, how the methods have been called at the time, was to exploit the entire information used in the images. Because as you see here, what the pioneering groups have exploited was only a single scalar per image, right? It's only the photon number, and therefore you calculate only one ratio. Yeah, it's not the full information contained in the image. So if you take a rather holistic approach, which we did by using an MLE approach, using a correct model for the PSF, you're also using information that's buried in the shape of the point spread function. And you can theoretically calculate what's to expect using these gamma Rao lower bound calculations, and these curves can you see here. If you only use the ratio, you end up with something such as this, a dashed line. And if you use the entire information that's contained in those two images, you end up with something that looks like this. This is the solid line. And you see at the maybe in the first 50 nanometers or so, the information is dominated really by the intensity ratio in the channels. But from then on, the shape takes over, because then the molecule goes a bit out of focus, and then the shape contains the information. The ratio doesn't contain a lot of information anymore, and you end up with a much nicer behavior of your information curve. However, there's one problem with that. If you want to draw information from your PSF shape, it needs to be super well known, right? If you don't know exactly what your PSF looks, you're creating a lot of errors, which I will address then in my second part of my talk today. And our approach to solve this was to use basically phase retrieval. And first of all, we use a good model for the PSF, which is based on vectorial light propagation and Fresnel coefficients on the cover slip side. But then also, this is not sufficient. Yeah, you always have some aberrations in your system. Your PSF will always look slightly different. And to account for that, we applied a technique called phase retrieval. It was also introduced the first time 2004 by John Sidat's group, which is basically means or comprises measuring a bead, typically a sub-diffraction limited fluorescent bead at several planes, and then it basically fit phase aberrations to what you're actually seeing in the pupil. And this is what you see here on this slide is an exemplary experiment. Here you, for instance, see in the first row of images a theoretical calculated PSFs with the focus values of -1200 and +1200 nanometers. Okay. Then in the second row, you see how it actually looks in the experiment, and you already see, okay, there are really some differences here. It looks much smaller and also a bit different. Brightness distribution is different. But then after adapting a bunch of Zernike modes using this phase retrieval method, we see that our adapted model pretty much resembles what we actually see in the experiment. And you see here in this bar graph, what we found, these are aberrations that we typically see in objective lenses. These bars are actually pretty small, right? So all of these phases here, yeah, this y-axis, the phases, they would be considered diffraction limited. Yeah, the diffraction limited would be here in these units at around 0.45, right? And these are pretty small altogether, mostly spherical aberrations, but they nevertheless kind of change the PSF quite significantly. Okay, that was one thing we wanted to do to make just the most out of the information that we have. The second thing was, maybe can we do it more simple? Because beam splitting, it sounds easy, but if you just have a microscope and you have a biolab, you don't even want to do that, because it means you have to introduce beam splitters, you have to use camera ports, and then the beam paths are not really optimized for that. Could it be, or is it possible actually to basically reduce the setup to something that's as simple as possible? Just use a single channel, but use the same information. And the first idea that we had was, well, maybe you can use kind of a phase shifter in the back focal plane like that, which introduces a phase shift to the SAF light with respect to the WAF light, maybe pi, because if you have a pi phase shift, then whenever SAF light appears, it will on the axis destructively interfere with the rest of the light, with the undercritical light, and then maybe you get, by like changing the interference conditions, a very sensitive PSF. And that works, right? So the simulation here shows that the molecule here at 2 different positions, 0 and 200 nanometers, which are pretty hard to distinguish in a standard microscope. They show a clearly distinct PSF change. However, we never put this into practice. Why? Because we pretty soon figured out that you can do it much easier. And this brought us to defocused imaging. It turns out that the only thing you have to do is to slightly move the objective lens by 400 or 500 nanometers towards your sample. And then you also introduce a phase curvature in the back focal plane here, which does the job for us, right? We have a phase shift between soft light and rough light, and this changes the interference. Well, of course, it's a defocused image, and that gives you the same information as the pi phase shift. It's even slightly better. Off-focus imaging, however, I have to note, is not really new at all. Also, Jörg has introduced it into his talk today. It has been used to measure dipole orientations, because fixed dipoles, if they are tilted, especially if you collect the SAF component, has a drastic effect on the PSF. But also it has been proposed as a method to track molecules. As in a defocused manner, you can nicely track and follow with your defocus a molecule that diffuses around. Okay, so, well, having this decided on this method, we were also calculating what we can theoretically expect from it. And we were again calculating Fisher information-based Kramar-Rao bounds or precisions. And what we see here is in red, Salm or Donald. Yeah, so the dashed line actually is Donald. The solid red line is actually what we denote here as Donald or Salm plus, which is this more holistic variant of Donald. And the blue curve is a simple single channel off focus detection, right? We see, of course, here XY suffers a bit if you are out of focus because the PSF gets a bit broader and therefore it gets more easily buried in the background. But it's overall in the measurement range that interests us, not really worse than Salm or Donald is. And in Z, it's actually even slightly better, because we don't absorb any light. As you've seen in Salm or Donald, it's always based on also absorbing maybe a quarter of the light, which makes it intrinsically a bit worse. So it seems to be quite promising. What we also did after these calculations, we tested it just to see how accurate is the method. Are there any significant biases that we see? For that, we dyed a ball lens and just measured it using SMLM, and we see pretty nicely follows. So each dot here is a localization event, pretty nicely follows what we expect. So the red curve is the ground truth curve that we expect. We see a little bit like a trend to lower underestimated Z positions. Bear that in mind because this is actually a bias that we think we can explain. And also then, what about the precision? For that, we imaged a mesh of microtubules that is in a fixed COS7 cells. So cells build these protein tubes, these microtubules, which are pretty small in diameter, like 20-something nanometers in diameter. And what you see here is basically a localization map in XY, and the color encodes the Z position. Yeah, so this is in nanometers here. And it turned out that our precision was isotropic, and it was pretty good actually on the order of 10 nanometers in X, Y, and Z. Yeah. Also, because we used a very high NA here, it's 1.7. That's the highest you can actually get, and that pushes especially the Z precision pretty largely. Okay, so this method that we basically developed was then transferred to the Vienna lab, to Gerhard's lab, and I've seen Lukas has already gathered really beautiful results. So we are really happy about this. And congratulations. So it's now applied to the biological questions. For us in Innsbruck, it was time to concentrate on other questions, and actually two things we are interested in. The first, what about robustness? Yeah. It's also what we learned about when we looked now more into how real biology experiments work. It simply has to work, not 1 time out of 5. It simply has to work. So robustness is a really big thing. And what we did then was to investigate systematic errors that are pretty crucial in SMLM, and also to answer the questions, actually, can we somehow avoid them, or can we at least reveal them from the algorithmic side? Yeah, because if we can see this detection or this localization event is erroneous, we can actually kick it out, and then at least no harm has been done. And the second question we've asked was, what's actually the performance of other established methods close to the cover slip? There are a couple of comparative studies out there where we compare astigmatism to double helix to biplane, whatever, but those studies usually don't even include this sub-effect. And no single one of these studies was actually concentrating on our application target working range, which is the first 200 nanometers over the cover slip. And we were investigating biplane imaging. I've mentioned before, it has been developed actually to stretch the working range. Why did we consider it here? Because biplane imaging actually has one defocused image, such as defocused imaging does. So it's supposed to have a really good C precision. And the other plane, that image, can be taken in focus. And then it can maybe restore the XY precision that suffers pretty much in defocused imaging. And astigmatic imaging has also a defocused component. This is the defocus there is only applied to a single axis, but nevertheless could be promising. So we did a systematic comparison, therefore, of defocused cylindric lens and biplane imaging. Yeah. So all these methods are sketched here. And all of these methods have certain operating parameters that can be optimized, right? Defocus imaging has only this defocus parameter, a single parameter that we call here gamma. How much do you detune your objective lens? Cylindric lens imaging has the degree of freedom that you can defocus it as well, but you can also introduce astigmatism. We can introduce varying astigmatism by a pair of cylindric lenses, and the parameter A6 here is basically the magnitude of the Cernicke first order Cernicke astigmatism. Yeah. So the null in the null scheme 6 means is just the number of this mode. And in biplane imaging, we also basically can both channels defocus independently. So we decided to define a common defocus gamma, same definition as in defocused imaging, but then also a kind of neutral additional defocus in one channel, which is called here capital delta. And we basically optimized brute force optimization, all of these techniques for the working range 0 to λ/3, and then just empirically could derive a formula which allows you to make a choice of these parameters that give you the optimal precision in this working range that you see here in this table. Yeah, that's the result. So that's just the obtainable, maximum theoretically obtainable precisions. They are performing pretty well, but what we see is slightly the best method is biplane imaging, right? It has maybe 30% better performance than defocus imaging, but it has also the best, at the same time, the best XY localization precision. But overall, they are not so different, right? It's not that one method would be 2 times better than another method. Okay. So, of course, we wanted to confirm this in experiments, and to do that, we used a ground truth sample that we think we can trust, or that we actually now turns out that we trust. And these are DNA nanorulers that you can actually buy from DataQuant. These are actually 80 nanometer long DNA strands bound to a cover slip, and it's based on DNA paint. So you have other strands that are actually linked to a marker that stochastically bind to these fixed strands, which actually can be oriented at different angles, like here in this artistic sketch here on the right. And this is how the raw data looks. Yeah, and for any of you who have actually done experiments in SMLM, will agree that this is a really beautiful image. So high signal, low background. So it's actually quite good to work with. Also, we were sharing the same basic imaging platform with all of these methods. So identical excitation conditions, objective lens, and so on, also tube lens. The only thing that we changed was we could flip in our cylindric module here to do astigmatic imaging, and we could flip up a mirror here after the tube lens to relay the system in order to do biplane imaging. But the basic layout of the imaging platform is identical for all of these methods. And these are the results. So what we see is here in the first row, each dot is a nanoruler. So this is actually the image of a nanoruler. And if we have that image, we get basically a cloud of localizations on the marker on the one end and on the other end. And these clouds allow us to determine the length and also the angle of this nanoruler. And according to the length distribution, we find that we are pretty much there what the manufacturer says. Yeah, the manufacturer says that the length distribution should be somewhere between these red dashed lines, and this is exactly where we are. Also, we see a lot of different angles. It should be pretty equally distributed. What actually is apparent that in the focused imaging, these upright standing nanorulers, they tend to miss, right? You see basically a hole here, which means there seems to be a problem in actually identifying nanorulers which are standing upright. And then if we compare the experimentally obtained and theoretical precisions, we also see a pretty good match. So the black numbers here in nanometers are the theoretical precisions. So we have around 4,000 photons per localization event. And the red ones are the experiment. We are pretty much there what we actually think would be obtainable. And again, we see that biplane imaging is slightly performing better than the other methods. So the next step is about systematic errors. Okay, and there are actually three main systematic errors that can mess up your measurement. The first are aberrations that I will address here. The second one are dipole effects, as we've seen beautifully today already. If you have a fixed dipole, it simply doesn't look at all like your model image. And the case of a double emitter event. Yeah, if you don't recognize that what you have seen have actually been two molecules, then you also end up with a systematic error. And in terms of aberrations, we have actually three types that are important. Spherical aberrations that are really a pain for microscopists, and also astigmatism and coma, which are field-dependent aberrations and therefore pretty important. Now, what you see here in this graph is the impact of sub-diffraction limited spherical aberrations on your localization coordinate, axial localization coordinate. And you see this range here goes from -72 to 72 millilambda. That's the magnitude of the aberrations that we have. Why this strange number 72? Because this is the Maréchal diffraction limit. If you buy an objective lens and the manufacturer tells you, oh yeah, it has diffraction limited performance over the entire field of view, then the designer accepts aberrations within this range, right? Because you always have to make trade-offs. So you can add up with aberrations such as large as this, right? Very easily. And you see here from the PSF images that these are actually not really big aberrations. The PSFs, that's the XZ image, doesn't seem to be affected so much at all by these. Nevertheless, we see that spherical aberrations of that magnitude can introduce really big errors, up to like 50 nanometers easily. And that's a quarter of our measurement range, right? So this kind of emphasizes how important this is. We did the same for astigmatism and also for coma. This is, as I just mentioned, the important three. And we see all the methods suffer to some extent. What we can see here as a very overall pattern is that each method suffers most at the aberration, which attacks the source of information for this particular method. Cylindrical lens imaging draws its information from the ellipticity, right? So from astigmatism. But if you have a bias of astigmatism, of course, you have a very big error. Same with defocused imaging. It basically lives from defocus. That's the source of information, which is a spherical aberration. In turn, it means if you have another source of spherical aberrations in your system, it will react very sensitive to this. What we can also see is that biplane imaging seems to be pretty robust compared to the other ones. Okay, so like these biases might in the end not be so bad at all if we at least would have the ability to identify these biases. If our algorithm says, okay, here we have spherical aberrations, and then a red warning sign goes up or something like that, then maybe it's not so bad at all. So what's interesting to see is how, with the p-value test and our model, how likely is it that these errors remain unnoticed. So what we did basically is we calculate for each event a goodness of fit number, the log likelihood ratio here, and that should be chi-squared distributed like that. And then if it's actually something wrong, then the LLR value will be larger than that. And then we can do a p-value test, set the threshold at, let's say, 5% or 20%. And everything that's like here for 20% LLR value above 310 or something, then we just throw the data away. And that's basically summarized here. Here we see mean LLR values for varying aberrations. And what we see here is dashed lines are 5% or the even stricter 20% significance level. And what we see here is that, for instance, if you do the focused imaging and you have spherical aberrations, then you have no chance to see them, right? So your algorithm will be blind to the existence of this problem. Same situation actually also for cylindric lens imaging, also for biplane imaging is a bit better. Well, you can discuss on that in detail if you wish, but I think it is the take-home message here is basically that's simply not easy to identify even small subdiffraction limited aberrations. I think this is what here the message should be. Yeah, systematic errors is another problem. Could be a problem, or you can also, as we've seen also today, could be also a source of information. But here, if you have a model that's based on isotropic emission and you get images like that, then it's a problem, of course, because you see these images have nothing to do with this model. And you see in particular these 45-degree tilted dipoles, it kind of has a move, a center of mass moves to the right here, right? And especially here you see that in this angular range, you get a lot of lateral bias. That is pretty well known. And you also see that the bias is pretty high for most of the method. But what's actually pretty nice or apparent is that biplane imaging is really an odd man out here. It seems to be much more robust compared to defocused and cylindrical lens imaging. So if you look a bit closer into this, why that actually is the case, then you see that both channels in biplane imaging individually, they are strongly biased, but it happens that the bias goes into opposite direction. So we are really, this is a pretty lucky situation. They overall, over the range, they pretty much average out. And this is the explanation of why biplane imaging is pretty robust to these dipole effects. Again, the question, can our algorithm identify fixed problems related to fixed dipoles? And well, the answer is, if the dipole is really immobile, then it's pretty easy. That's good news, right? So it should actually be apparent to the algorithm and you can kick them out. But also I have to mention that a fixed dipole is, so to say, the worst case scenario. What's much more realistic is to have a mix, like a slightly axially wobbling dipole and so on. And then of course the situation will be different than shown here. So last but not least, what about double blinks? Yeah, so multi-emitter events are known to be an issue for SMLM, and we made kind of a more systematic investigation here. We were assuming images like that, basically 2 emitters in an image at varying distance d, ranging from 0 to 250 nanometers, and then for different c coordinates, calculating these bias maps. Yeah. So you can do that for the focused imaging. For cylindrical lens imaging, you need actually to take into account 2 different cases because of the anisotropic PSF. You must actually consider lateral and vertical or lateral distances here, distance vectors d, which actually changes the situation. And also for biplane imaging. And what we see here is that, and this is pretty intuitively explainable, in defocused imaging, like the appearance of 2 emitters next to each other, it leads to an underestimation of the C coordinate. Why is that the case? Because 2 emitters, they look pretty much like a single emitter that's more defocused. And defocus is the source of our C information. And therefore, it's clear that we end up with systematically underestimated coordinates. And we believe that this is actually what's happening here. If you remember this characterization measurement, that if you have double blinks, then you end up with localization events that fall down to lower Z values. And also this is the explanation for the upright standing nanorulers, because if you have events that fill up the gap between the two clouds, yeah, in the nanoruler, then your clustering algorithm just fails to identify two individual clusters there. So the last slide, or almost the last slide, is how easy is it to identify double blinks? And there, of course, it depends on what the separation is. And what we see, that's actually bad news. If you concentrate on the blue and the red curves, these are separations of, well, 0, then you can't distinguish it, or 185 nanometers. And in these cases, you simply cannot see that you have a problem. Yeah, that this is actually a double blink event. If the separation grows larger, then of course it gets increasingly easier. Okay, to round up the talk, so I introduced you to the focused imaging, that this can actually be a really simple but powerful, nonetheless, method to obtain high precisions close to the cover slip. And I also provided a comparison to other methods that are well known, like biplane astigmatic imaging for this application case. So we found optimal imaging parameters. We compared also precision and robustness. Actually, what in the end the conclusion should be, at least what we found, is that if you can cope with the additional complexity of detecting two channels and doing joint data processing, then you should actually go for biplane imaging. Okay, so last thing is to thank the group, like Philip, who recently graduated, also Lisa and Clemens in Innsbruck, and also the guys in Vienna. It's really a pleasure to work with you. It does work really well. Thanks a lot. Thanks also to the funding agency and to you for your attention. Thank you.
Thanks, Alex, for this presentation. Very interesting stuff. We knew it, of course.
Yeah.
Other questions?